thank you everyone for coming. I appreciate that. So this is part of our Institute for Digital Ag seminars. We have basically seminars. I mean, we are putting here the seminars that are coming soon. So I think that you all receive, if you don't receive these emails and newsletters, let us know. Melinda is here, so we can make sure that everyone gets emails on the seminars and newsletters. So just in case, I think that Dennis is right there. So Dennis Buckmaster is director of the Institute, and Ignacio Ampili, co-director of the Institute for Digital Ag. Melinda is also helping our managing the Institute. So, and today is a great pleasure to introduce our friends from Corteva. So I think that I will introduce first Byron, and then Byron will introduce everyone because we have many speakers. So Byron Slew, I mean, he's working on expert on the Biology Center for Corteva. And then today they're going to be talking a little bit about digital ag, predictive decisions on crop protection solutions. So Byron, I will just let it so you have time.
Thank you so much, Ignacio. Good morning, everyone. Can you hear me okay?
Yes.
All right. Melinda promised steak lunch, and I think that's what brought the crowd in here, Melinda. Thank you for advertising for us. We have really been looking forward to this for the last several weeks. And Sam, thank you so much for reaching out and helping us to set up this opportunity to visit with all of you here today. You're our nearest neighbors in so many respects. So today, the Corteva Group is excited to talk to you, and we have this title here, Designed for Purpose. And there's a great reason why you'll hear us use probably the term design quite a bit, because very often people talk about, oh, you discovered this molecule and you did this. And yeah, there's a lot of work that goes into what ends up with our customer and years of actually designing it to meet the need for the customer. So it's not just, you stumble on something and 12 years later, it's the same thing that goes in a jug to the customer, as you'll hear from our teams. And so what I wanted to do, just very briefly, and as you hear from the speakers, you'll get a little bit more on their background, but we'll have Phil, Sean, Ken, and Lucas tell you a little bit more specifically about the areas in which they work and how that actually enables us to do what we're wanting to do, which is to get all of these solutions to our customers to enable agriculture to continue to be the source of not just abundant, but secure food supply for everyone. All of us work together in different ways. My teams interact with all of the others that you see in different ways, and you'll see how some of that come out as we go through today. So my simple role just for the first couple of minutes is just a storyteller to tell you why what you'll hear from the other speakers is so important, okay? So what drives us at Corteva is really innovating for the future of farming. And you just need to be around agriculture for 2 minutes to realize agriculture today is not even what it was last week. That's how fast technology is moving in our industry. So we are about innovating for the future. We're also about securing food and energy for the future as well. If you've been paying attention to Corteva, you've probably heard about one of our latest joint venture that includes looking at canola oil for use as energy, and then preserving farmers and their livelihood. As we talk about those who consume and those who produce, agriculture is not just about the farmer. The consumer is what makes agriculture what it is for us today. But you don't stick around with farming the way it is now if it's not something that goes far into the future. If it's a one and done kind of thing, and we hear about fewer farms going out of business and all of those kinds of things, we're about making it work for the farmer, because if it works for the farmer and the farmers are consumers too, then it works for everyone. And then we're also wanting to do this in a sustainable way. We all have a responsibility to make sure we maintain this planet that we live on and try to make it continuingly habitable for all of us. So this sort of seems like an easy thing to do, but I wanted to set this up. Our customers are very exacting in so many ways. They want to make sure that whatever they're using, you have lower and lower use rates. And you see that in what's coming in the industry. Many of the products that are there, and we have a lot of those extremely low use rates compared to older products. They want new and novel modes of actions, and we have to do that because resistance is an issue for reality, whether it's weeds, insects, diseases, it's there. They want new and better formulations, and you'll hear from Phil as he talks about how we use data to do some of what we do in designing experiments. You'll hear from Lucas about the challenge with resistance and some of the things that he does as he works with the mode of action group as well. Natural products becoming really more important, and that's a very important thing for us. We have a significant amount of work in this area, and you'll hear from Ken as he talks about this. They want these solutions that they get to be durable, long-term. They don't want to have to switch tomorrow if they don't have to. There are active ingredients that we use in our crop protection business that are 50 years old, and they're still functional. They'll still do what we want them to do, even when you still have some resistance issues that are there. And everybody wants an easy button, and they want it to be easy. All of this is awesome, and it's great that this is what our customers would like. But the reality we live in is as we try to go from discovering to launching new products, you see the roadway and what it looks like. The one that's a straight shot, the bridge is out. Okay. You have an off ramp that could work really great there, but really on the top there, that's the reality in which we live. And really that reality is based on a lot of the regulatory hurdles that we face. And as a result of that, what we do, and these numbers we get, that these are industry averages, but they're not far off. I can tell you as you go company to company. The journey is about 12 years and about $300 million for each active ingredient from discovery to the first time a customer is able to buy this. So what's the challenge, and why are you here today to hear from our colleagues and to see yourself in this journey in the future is because it's not getting cheaper. The time is not getting any shorter. So what can you do to give yourself an advantage if this is your reality? You've got to learn to do things quicker, better. You've got to learn to do things in a way that will give you an advantage because competition is ever present. And that's what my colleagues will share with you today. And I hope you see an opportunity for you to be a part of this. And candidly, even if it's not with Corteva, we still need you to do this wherever and with whomever you're doing it, because all of us have a dog in the fight here, which is, like I said, we all eat. If we can agree on that. So it's great to be here, as I said, and I'm looking forward to our conversations even after this is over. Last slide, and we're going to switch to Phil. As you look at this, many years ago, it took way more shots on goal in a way before you could say you were successful, and it took longer. It was cheaper to do it as well. Today, we're screening tens of thousands of molecules before you end up with one that would go to a commercial product. But again, to go back to my previous point, we're able to do this because of a lot of the high throughput screenings that we have. And so you will see from the stories, regulatory clocks, they do what they do, hurdles don't disappear, and pests are ever evolving, and we have to keep up with that. That's the wicked problem that we face. So with that, I said Phil. Yeah, Sean's up. And so with that, I'll pause there and go in the wrong direction on their slide there. And we'll have Sean come up and talk about faster, better decisions. Yep. Sean's in regulatory, so he's used to the hurdles. And that's why he's sitting over here.
Yes. Well, thanks, Byron. Well, thank you so much. It's really wonderful to be here. Thank you for the invitation. So my name is Sean Guillen. I have a background in toxicology, so not really in agriculture, but what I want to talk to you about is our capability related to predictive science for kind of early stage assessments that we do. So, and this is kind of building on what Byron shared, right? So this is the, kind of 12 plus years to go from kind of a lead, a concept, all the way to a product that we can launch for our customers, and that $300+ million of investment. And the interesting thing is, like, that investment, it's not linear. Like, it really, really ramps up in development. That's where we really start spending a lot of resources. Yeah, and why is that? Like, why is that so challenging? Part of it is the regulatory path and journey that we're on, right? So, to get from here to a product that's in our customers' hands, we have to traverse a lot of challenging territory. We have to understand, how the molecule behaves and interacts in the environment. From an environmental fate perspective, we have to assess impacts on non-target organisms such as pollinators and aquatic species. And then there's the whole human health assessment part of it, toxicology, human exposure to residues, and safety to workers and applicators. And all of that has to go into our dossier, what we submit to the regulators. And then they will evaluate that and go through their process for approval. And that regulatory bar continues to go up and to become more challenging, right? So there's a lot of areas where we can kind of fall off this pathway and end up with potentially spending a lot of time and resources on something that ultimately doesn't get us where we want to get to. So that's where our investment at Corteva in predictive safety really comes in. So what we do is we invest some resources very early on in this process in a very focused manner. So what this allows us to do is kind of de-risk our pipeline. So we kind of refer to this as a fail fast, fail cheap kind of approach. So if we're going to identify a potential issue or challenge with registerability, we'd rather do that here rather than over here where when we've invested that $200-300 million. So the idea is we want to identify these issues, resolve them as early as possible. This gives us more options, the ability to pivot, look at other opportunities, that kind of thing. And ideally incorporate those learnings to help inform the actual design of our molecule. So the ideal molecule in our mind would be something that we put out into the environment. It does what it's supposed to do, and then it dissipates, and it doesn't have any impact on non-target organisms or human health or anything like that. So that's kind of what we're ideally working towards. So, the objectives here are to lead us towards regulatory success. And then, as Byron touched upon, making sure that our portfolio at the end of the day is sustainable, that the products that we're putting out there. So, I'm not going to go through all of this in detail, but big picture, this starts with us using a variety of high throughput approaches. So in vitro methods, cell-based assays, a lot of in silico modeling, and then some focused whole organism testing applied very early in our pipeline. And we're looking at a number of different endpoints. We're looking at environmental fate, toxicity, metabolism, and those sorts of things. And then as we progress through the pipeline, we're basically, kind of doing higher tier, more definitive versions of those types of methodologies. And all of this is, at the end of the day, really designed that we are, increasing our confidence that, this project, this molecule, this analog that we've invested in is going to be able to go the distance and, be registered by the regulatory authorities. So this is just sort of a map of what we're focused on in my team, the Predictive Safety Center. So, we've got a couple of big picture items that we're looking at. One is human health. And, some of the endpoints of interest here are things like carcinogenicity, developmental reproductive toxicity. So what do we do to kind of understand those endpoints is we use a suite of higher throughput methods and assays. So one of these is a high content imaging approach that we have called cell painting. So that's a phenotypic assay that looks at cell health in a very high throughput manner, and then we can take that information and help use that to inform the decisions that we make as a project team. So that's one example. Another is transcriptomics. So looking at gene expression changes with treatment with a chemical of interest. For environmental safety, we do a lot of similar approaches, but we also have several different organisms that we look at and assess within our team. So these could be aquatic organisms, they could be non-target plants, pollinators, things of that nature, where we can get an early indication of potential impacts and effects in the environment. And then underpinning that, we rely a lot on laboratory automation, a lot of analytical support, and all this data at the end of the day kind of feeds into our modeling and analytics platform so that we can build models and become more predictive over time, hopefully with the need to generate less data. So I think just to kind of wrap up, we really believe strongly at Corteva that this early investment in understanding human health and environmental safety is really important. It helps us de-risk our pipeline, reduces the potential for kind of that late stage attrition in our pipeline, and ultimately helps to ensure that we're bringing forward product concepts that are going to be favorable from a human health and environmental perspective and ultimately sustainable. I think one kind of proof point of, Corteva as a company, our, focus and, investment in this area is really manifest through the fact that we are a 6-time recipient of the Presidential Green Chemistry Award. So these are the different molecules for which Corteva has won that award over time. So really kind of highlights that this is something that we've been invested in for quite a long time. Yeah, and I'll just mention collaborations are very, very important to us. We are, incredibly interested in, working with different organizations across government, academia, and industry, where we can learn about cutting-edge predictive approaches, whether those can be applied in the human health or environmental space. And these are just a few of the different collaborations that we have going on. So with that, thank you so much for the time. And I don't know if we are holding questions till the end. Is that the plan? Or?
Yeah.
Okay. All right. And so I guess I hand it off to Bill.
Well, it's a real pleasure for me to be here. So thank you for welcoming me into the group here. One of the things I'm very strong believer in is diversity in all sense of that word. And what I noticed today coming into this room and also looking at the great agenda that was set up to us is the diversity of skill sets that it really takes to serve agriculture. And I'm probably unique in the room that I'm not a biology person, I'm not an ag person, and so I'm coming in as a chemical engineer. And so I spent most of my career working on product development and the chemistry that goes into serving our farmers with awesome, good ag products. So I was trying to think of the audience here today and like, well, how can I maybe say some words that maybe could be of some value? So humbly here, we'll see if this hits the mark or not. But in the space that I'm going to talk about is digital transformation. And so we see it everywhere in our environment. Even our cars now have, all these screens on there and all this. And so what does that mean then as we run our science? So maybe with a raise of hands, who takes most of their notes these days with a paper notebook? Paper notebook, paper notebook, paper notebook. Okay, who takes notes now digitally, right into their laptops? Okay, look at that. So I was actually making an observation this morning, Byron, as we came in, and I scanned it as our Corteva team was taking notes and talking to the Purdue team. Look at this. I'm leading digital transformation for my lab, and look what I got. I got paper. But my colleague Sam Zukowski, he has his OneNote open, and he's taking all his notes in OneNote. Now we can envision whose data is going to be more valuable as we leave the campus today and say a year from now. One thing is, I am totally unorganized. This notebook is going to be in a drawer somewhere, and I got 5 of them for all different ways. Sam's going to have his data digitally captured. So now he can go into Copilot or any of those things and say, what did I do at Purdue that day? Question mark. Bam, he's going to have a summary of that information where I'm going to be fumbling around looking for my notebook. So anyway, gone are the days of the paper notebook. And so while I'm still very much paper, this is one of the challenges of the digital transformation is I am a Luddite. I am back in time, but I see the future. So my daughter is in 7th grade. She is no longer using paper for any of her assignments. Every one of her assignments is electronically entered. And then I try to get on the computer. She asks me help. I am lost. Like, where do I even, sorry, my it gets kind of excited. See, maybe my phone or even my watch is giving me status signals here. So anyway, gone are the days of this. But when I started my career, this is what I had. And I physically had to print off graphs and tape them in. I had to tape graphs in here. How ridiculous is that? And I had to initial by them that I put that in there. So somewhere there's some notebooks with my name on them, maybe archived. But is that data valuable or helpful? Now, of course, it's probably scanned. So I'm going to talk a little bit about how we've been trying to transform all of our labs at Corteva for digital transformation. I talked to one of your colleagues. One of the things that's really important is making sure we have a nomenclature for how we talk about things. So even within Corteva, as an active goes from that discovery stage all the way up to now, that name changes 5 different times. And so as I tried to collate and bring our data together, some of it's like data matching and trying to name stuff differently and then trying to get that structure to it. So I am convinced the lab of the future is going to have a data or have a scientist with lab automation, modeling informatics, and data and knowledge management. So it's kind of like all 3 of those tools. If any one of these pieces is here, we're going to fall behind and not advance as we are. So the mindset we should all be having is having a fair data mindset. Making sure your data is findable, accessible, interoperable, reusable, and in our environment secure. So we don't want our data in the public nomenclature because it's our IP, it's our trade secrets. It's very critical to all of our stuff. And we want that data then to provide knowledge and then knowledge insights and ultimately make the right actions for that. So one of my goals this year is to try to use the new digital tools to get away from paper and try new things. So I generated these images with Copilot AI. I gave it prompts and I did it in 30 minutes. I thought that was pretty good for me. But it did make some typos, and I kept trying to make the prompts fix the typos, and it wouldn't. And then I guess got aggravated, and you're going to see typos in these graphics. So anyway, this is what the lab of the future looks like. And actually 3 of these things were not available when I started my career. We had very limited lab automated equipment when I started my career. We did not have data scientists sitting next to me, and we sure as heck didn't have AI LLM. So again, now these are going to be the things that you guys are all going to be doing now going forward is these sort of things. So I was just doing samples in little beakers, writing it in my paper notebook and doing my work there. But this is the lab of the future. And so as we think about a lab of the future, we have the platforms that we need to develop and use have to be considerate of who generates the data, and there's also consumers of the data. So any lab information, and Byron, how am I doing on time? I'm good. Okay. So for the teams generating the data, we need to be thinking about how do we make their lives easier and helpful. So we need to think about, what do we have labeling systems for them? What kind of material IDs are they going to capture their information to? Forms and documents and track the task and everything else. And then also ultimately, data is going to be coming from instrumentation in the future. So how do you structure that data and whatever else? I thought Copilot was pretty cool because we use X number nomenclature for material IDs within Corteva. I did not tell it that. Maybe it was just happenstance, but it generated X numbers as our material IDs. So I thought that was kind of funny. I have no idea how it picked X numbers, but that's what we use within Corteva is X numbers. So again, whenever there's really high quality structured data, then it's easier for the data scientists and the chemists to use. But this is not necessarily easy to find within Corteva. And what we find is scientists collect their data in a lot of different ways. So in our formulations lab, we have scientists in different laboratories, again, even maybe naming stuff differently by the actives that they're studying. And we don't have a structured way to getting that. So that's what we're trying to change is we're trying to develop ways that they all can capture it similarly. But one of the things I started, and when I started the project, this was like my pillar. Everything has to be structured. Every piece of data has to be structured. But I have evolved quite a bit because I really believe unstructured data sets also now with these large language models and the queries and the questions you can pose. I feel like now this is almost as important too. So I talk to the chemist and I say, hey, how are you going to structure your hypothesis into your data notebook? How are you going to, your observations that maybe are more difficult to structure? And you can imagine asking questions to that data and also getting results back. So Sam Zukowski is going to have a one notebook page today at Purdue of all his ideas and everything. Those things you can ask questions to in the future and maybe pick out something then to help solve your problem of the future. So these models, though, need to be able to go into different places within. You have some data in the experiments, you have others in databases, you have others in reports. So being able to search all across that is very important. And then, really the scientist is right at the center of this because you're going to be the ones collecting your data. So you want to make sure how can all the teams, think about structuring their data and also having these systems available, findable, accessible, interoperable, and reusable. And so then, if that's met, then a scientist anywhere in the world then can put in some identifiers, find all the data that we have on it, and then start making new hypotheses, new ideas, and everything else. And then the data consumers of the future is as much the data scientist as the lab scientist, really working together with our computer friends there. And so it's really an entire ecosystem here that needs to be together. So that's it. Thank you.
All right, so my talk actually resonates pretty well with Phil because Phil is the guy that usually makes me happy because I'm a data scientist at Corteva and he's the one that makes the data accessible and ready for us. All right, my name is Lucas. I'm a data scientist at Corteva, but I'm actually a weed scientist by training. I came from the greatest program at the University of Illinois. Sorry, Brian. But yeah, so I'm also a computational biologist by training, and I'm gonna bring the buzzword for you guys, artificial intelligence or AI, and how we're basically using that to advance our crop protection pipeline, both at the mode of action discovery and also down into the pipeline. How can we maintain our products in the market by tackling the resistant management? So yeah, what we've been trying to do. So my focus is highly into the genomics part of things. So over the last few years, we've been sequencing a lot of genomes from the enemy. So the enemy I call the weeds, the fungi, and the insects. It's where we want to know in deep how they behave, what is the biology behind it, what is the genetic diversity. And for that, we've been generating very high quality genomes for those target species over the years. And that can lead us to create a very in-depth knowledge about the genetic diversity. And that really creates a huge foundation for discovering novel molecules, driving insights about where should we tackle, where should we go, so we can deliver novel tools to the farmers. Okay, so we can do all this sequence. Sequence is easy now, but understanding that is the actual bottleneck. For instance, there are studies showing that you can sequence an entire human genome with $200 now. So it's cheap, it's accessible, we can sequence a lot, but the bottleneck now is how can we design the understanding from that that can lead us into discovery and decisions. Because we need now to go into the pacing that we are generating this data. And the other thing, when you think about discovery, it's not a single model thing. It's not like a yes or no question. We cannot have a model to rule them all. We need to think about like a decision of chains. It's a reasoning step. So we need to thinking about how can we use AI, for instance, to enhance the scientific reasoning process. How can we help our scientists to actually tackle down all the data that we're generating into a more sophisticated in creating new tools for them to use it. And that's where AI comes into play. Okay, so why is it so difficult to make discovery of novel crop protection products? So here it's a little bit about our discovery process. Phil and Sean already touched that. We started with many ideas, like, oh, we could do that and this and this. But as we go into the process by looking at safety, by looking at the targets that we're trying to hit, we end up with very few winners. And this is a very long process. It takes many years for us to put a molecule in the market. And we need to balance efficacy, selectivity. And of course, it's a race arm against nature. Resistance will always be there. So we need to be smarter than nature. It's tough, but we need to do it. How can we use data to actually go into that and be more proactive to deal with that? So my work actually goes all those scopes, and we are trying to use AI to actually first reduce discovery timeline, and second help our farmers to actually have the products that we're putting in the market for a longer time. Okay, so what we need to have to actually build those AI systems, it seems very cool, but it's pretty tough to do that. And as Phil mentioned, data is the foundation. If you don't have data, good data, your data garbage in, garbage out, right? So data, it's going to be the foundation. It's what's going to be like things like context reach, different data sources. How can we integrate that? How can we make like a fair approach for the data? So all of that is the foundation for everything. And the other part that I'm going to touch is models. What models can we use to actually drive meaningful understanding? Again, I bring one example here, which is what we call biological language models. It's like LLMs, but we use the grammar of life, which is DNA, protein, RNA has our language. And we use those models to actually drive meaningful information that AI can actually use to start answering questions about those patterns that they're seeing. And then, of course, it's AI systems, which goes into reasoning. There is a lot of talk about agentic systems now. How can we leverage different teams of agents to actually design like a hypothesis, answer questions? So let's start with the last glamorous part of it, which is the data pillar. It's what makes the biology AI ready. So Phil did a great job in pointing out how we need to make that discoverable, how we need to make definitions pretty clear, metadata, who owns this data, and not just a simple Excel file that it lives in somewhere else computers. Those things need to be centralized and accessible. And I like to use the 5 C's for AI ready data, which is basically the data needs to be clean, the data needs to be context rich, compliant, catalog, and cared for. So we need to maintain all of that before thinking about, okay, well, I want to use AI, but if you don't have good data, you cannot do that. So those are what defines the foundation for it. And then there is, of course, biological language models. So those models are very interesting because we can treat sequence genomic data as a language. So for instance, AlphaFold allows us to predict protein structures, which is massive. We can now predict any protein structure and use that into our discovery pipeline. Evo model, it's a very interesting one. It allows us to generate new sequences. So we can actually generate new proteins, generate new genomes even. So there is a lot of potential for that. And those things allows us to convert biology into numerical data, in very meaningful numerical data that helps us to identify patterns. And there are many potential applications like new mode of actions discoveries. We can use that to predict resistance. We can use that to integrate knowledge with other data types. For instance, all the field trials that we do, all the lab chemistry data that we have, we can all integrate by using those models. And then the last part of it, which is what people are using that, it's using that the AI system, in the case agents, to actually add the reasoning step into the process. As I mentioned, discovery is a reasoning process. It is not something that we just say yes or no, 0 to 100. There is a whole process to do that. And nowadays, AI, it's been so advanced now that allows us to create very specialist agents that we can tackle with very specific tests and make them work together to rationalize behind them. And this it can help us to rank hypotheses, create rank evidence, suggest new experiments to our scientists. And the goal here, a lot of people fear AI because, oh my God, that's going to steal my job. But that's not the goal. It's just we want to give scientists another tool to augment it and accelerate their goals and help them advancing that. And there is a lot of work being done on that. And we need to think about how can we put that into our day-to-day. Okay, so if I wanted you guys to bring something home, I would say like 3 points. Data is the key. Data, you need to have that ready to go if you want to apply new tools. Biological representation, we need to think about how can we represent biology into our world of data. So how can we translate how biology, nature is into our data? And then how can we scale up the scientific workflows? Agent systems are a great way to do that. So I see that as the next step for us to advance and expand our capabilities as scientists. So with that, that's what I have. It's Ken. Thank you.
Okay, I'm the last one. How are we doing on time? Good shape. Okay, cool. Okay, so I have the privilege of co-leading some of our natural product discovery work at Corteva. So there's been some allusions to that as an important area of how we do discovery and try and bring new solutions to farmers. So I'm going to talk about that. I lead some of our metabolomics efforts, and so I have a little bit of a metabolomics bias, and that may creep into my talk a little bit, but I'll try not to let too much of that come in. So this slide kind of just reiterates some things we've already heard. This is one way of looking at our early discovery to product pipeline. And I just want to zoom in really on kind of some of the things at the very start of this. We kind of have two big pillars of how we're doing our discovery of new opportunities for crop protection right now. One is focused on mode of action and specific target sites, and Lucas was, talking about that obviously. And I'm going to highlight a little bit around what we're doing around natural products, and to a little degree, even microbial solutions, where we're actually looking at either living microbes or inactivated microbes as solutions for protecting crops. So natural products, huge focus for Corteva. Why? What makes them attractive for crop protection? One big piece, actually, is that almost every mode of action for crop protection has actually historically been identified through natural products. Natural products evolve outside among insects, plants, fungi. They actually have an ecological context that's relevant. And so finding those and being able to apply them is very useful. And so it's a great probe for finding new modes of action. Although it doesn't tell you what the mode of action is, that's a different problem, right? And then another piece is sustainability. Sustainability is a very complicated, multi-factor topic, as Sean elucidated for us at the start. But one nice thing about natural products, they do tend to break down once you put them out there. And so that's one thing we want when we apply agents out in the environment. We want them to break down. Natural products, they're biologically made. They can be biologically broken down most of the time. So this is a huge area our company historically, and we've kind of been leaders in this, actually, successfully commercializing natural products. From about the 90s on, there's kind of been a big, I would say, drought of natural products being successfully commercialized, both in pharma and in ag. We've kind of been the standout exception to that. We've had two major franchises come out during that time, one set of fungicides, all related to this natural product up here, UK2A, and then subsequent sort of semi-synthetic and fully synthetic modifications on that molecule to make different types of commercial fungicides. And then a major franchise of insecticides based around spinosyn and spinosad. And so these are major pillars of our commercial portfolio to this day, and focus of a lot of the work we do. So that's great, but there is a problem in natural product discovery, which is that a lot of the low-hanging fruit has already been picked. And so when we go in to find new starting points, new small molecule starting points derived from nature, we wind up rediscovering things a lot of times. So this is adapted from an Onion article from back in the day. I think the original joke was about the polio vaccine, but I repurposed it to penicillin because that's a great representation of actually what folks in this field are trying to find new natural product starting points run into. Rediscovery of old areas that we've already tested, we've already deployed, we need to find new things. And so we're moving to a much more data-driven approach to this at the foundation of how we try and find new molecules. And so, one sort of term of craft we sometimes use for that, we call this metabologenomics. It's collecting holistically metabolomics and genomics data on the microbes to produce these molecules of interest, using that data together to try and gain insights of where we can find these starting points. And so we're going from a sort of very phenotypic, we're gonna test things, look if they have bioactivity, and then kind of try and figure out where the bioactivity is from, to a more precision approach of starting with something where we know there might be an interesting molecule there, and then working forwards. So what is this? I'm going to fast forward through some of these animations. What's this look like in practice and sort of foundationally? We have a really big microbial collection that we leverage. It's actually, in fact, I think these numbers that are on here need to be updated because we recently acquired quite a number more. This is actually very close to 1 million strains now that we have, proprietary Corteva strains in various states of cataloging and being cared for, I will say. But those are part of our collection. So we need to leverage that. So we subject that to both genomics and metabolomics at varying scales. Now we're not doing metabolomics on all 1 million of these. That is not within scope of the normal automation we can do. But we are doing about 4,000 of these a year, which is a really large scale historically for the field of doing sort of microbial metabolomics. And we have genomics on actually hundreds of thousands of these now for whole genome sequences. So it's really quite a lot of data. That hopefully lets us find new small molecule starting points that can go into our bioassays, that can go into chemical optimization if that's what makes sense. If it's going to be fermentation derived, it can go through sort of strain improvement and fermentation optimization to get us to commercial products. So one way to think about this is generating a digital twin of our microbial collection. And so this is one application actually that relates to finding bioactive microbes that we apply as crop protection agents, where we're actually looking for specific known genes that have some pesticidal benefits, and we just screen through our genomics data to look for that. You get like a matrix of the different features, pull out your candidate strains, and those candidate strains are going to be the ones that actually get prioritized and go into your empirical bioactivity testing. This is one thing that's really cool about ag, I want to say, compared to pharma, and a lot of like my original background was on the pharma side. At the end of the day, you get a great molecule in pharma, you cannot go and test that in people. That is very frowned upon. Not allowed to do that, okay? We get to go to the greenhouse, test it right away. I mean, as you all know and appreciate, but this is something that is a great advantage that we have, so we can actually get the empirical validation we need. And identify hopefully strains that have some of these useful bioactivities. focusing more on the metabolite side of things. Doing metabolomics on this many strains generates an enormous amount of data. And this is one of the things that's kind of held back, I think, the metabolomics field at times, is how you link all that data together. We rely really heavily on an approach called molecular networking, where you get a really high resolution MS2 mass spectral fingerprint of each molecule that's in these extracts from the microbes. And so this is a representation of what that global data set looks like right now. Things that match known molecules are characterized sort of in pink here, and things that are uncharacterized are in black. So you can see there's a ton of molecules there that have not been characterized. We don't know the structures. We don't know the bioactivities. So we think there's a lot of opportunities to go after. So what do these fingerprints tell us? Well, they are directly derived from the structure of the underlying molecule. So it can tell us about chemical structure. It can tell us about how it's biosynthesized. It can tell us about what sort of genes we should look for to try and find more strains that produce it. And so we've been deploying this approach. So one recent success story, of course, we can't share any real details about any of it. So it's just a very high level example. But for example, recent family of natural products that have some herbicidal activity, specifically on broadleaf plants, were identified through this approach on the basis of basically being things we hadn't seen before. We saw the mass spectral signature. We didn't know what it was, but we knew, oh, we haven't really looked at this before. Let's follow up on it. And so when we find a starting point like that, we then really get to use all the tools in our toolkit. We go back to the metabolomics and the genomics data. We're able to find strains that produce even more of it because we know what we're looking for. So we get improvements that way. We do optimization of the fermentation. We can get more fold improvement. And those improvements in how much you produce are really important to eventually being commercially viable because you have to be able to produce this at a cheap price point. And so that's kind of the next stage is further optimizing fermentation titers for something like this for it to be successful. So that's the conclusion of that section, and I will step aside.